So I'm working on B-cell acute lymphoblastic leukemia, which is a very aggressive type of leukemia that's the commonest cancer in children and is also seen in adults. And we're trying to develop novel treatments for that, and in particular put them together into new combinations in order to maximise the efficacy of those treatments and minimise the chance of resistance and toxicity. So this project stems out of some preliminary data that we have developed as part of another related project, and it relies on a technique called CRISPR screening. So what that does is it allows us to target every gene in a cancer cell and see which ones that cell is dependent on. And each of those genes could be a potential drug target. So we can actually look across thousands of different possible drug targets. When we got this data, we were really surprised because it turned out that all sorts of different types of ALL, this type of leukemia, were dependent on certain key targets. And when we looked to see which one of those we could drug, we actually found 25 drugs, many of which are currently used for acute lymphoblastic leukemia, but also ones that we would really unexpect. So we've now tested drugs, for example, that are used in breast cancer and found that those can be used in ALL too. What we wanted to do is then take that forward and try to work out new combinations of these agents together. So to do this, we've been doing some computational biology, using existing data sets from patients, looking to see recurrent patterns in those that might implicate potential drug targets. And we've set up a collaboration with the CRUK AstraZeneca Functional Genomics Centre to do a cutting-edge CRISPR screen, where we can take each of these targets, about 200 targets, and pair them together. And that gives us 40,000 potential dual drug targets that we can screen in one experiment. And then we'll take the biggest hits from that and take those forward for preclinical testing. Current treatments for ALL are, in children, they work, they're effective, but they're very toxic. We're talking about giving children lots of different types of chemotherapy drugs over 2 to 3 years. And in adults, the majority of adults can't tolerate that kind of treatment. In younger adults, we might give people high-dose chemotherapy and a transplant, and even with that, the outcomes are poor. We know that we need to find new treatments for this disease, and it's going to be very difficult to add those into regimens which are already quite toxic and quite complicated. So this project seeks to completely revolutionise treatment for ALL by moving us away from these traditional chemotherapy regimens into combinations of these new agents that are coming through all the time. And it will give us a roadmap of how different drug combinations can work in ALL, which we can take forward in the future. So in the first instance, this research will benefit the patients at highest risk. So those would be patients who relapse, older patients, including most adults, very young patients such as infants who tend to have very high-risk disease, and patients whose leukemias are driven by very high-risk genetic drivers. So all of those patient groups stand to benefit in the first instance. But if these things work, then we could potentially bring them into the front line across all patients, and that which hopefully will improve outcomes and reduce toxicity. It's an amazing time to be part of a clinical scientific team bringing this kind of research through towards patients. It's building on decades of research, basic science research that has moved us forward to a point where now we're at a position where we should be able to deliver, I think, within the next 10 to 20 years, real changes for patient care. And what I hope will be the outcomes from that is that we'll, as a clinician, will look at an individual patient, and by very precise diagnostic testing and response monitoring, we'll be able to tailor the treatments for each individual patient from a cupboard full of potential drugs that are coming onto the market at the moment. And we're already seeing that. So some drugs have been around for 10 or 20 years and now off patent, and we can use them like we would use antibiotics for an infection. We can actually just tailor them to the patient in front of us. And I think that's the way that cancer treatment is going to go more generally.